Good morning, Year 12, and welcome to another lesson of DNA, genes, and chromosomes. Now, obviously, it looks like we're going to be in this for the longer haul, so I would definitely expect that we can be keeping up with this work. Now, obviously, I'm speaking to those people who are watching the video. I'm sending some emails around to a few people who are not watching these videos. It is very important that you keep on top of this. If we're off for a prolonged period of time, obviously that will affect your learning. What I would value is people keeping an eye on what subjects they're struggling with and what subjects they're finding easy. Now, I would anticipate that translation and transcription are going to be one of those things that some people find more difficult. I may be wrong. I'd like you to keep a list of those subjects you found most difficult, so that when we do get back, hopefully after February half term, we can go over some of those subjects. I hadn't planned to do that, but with us being off for potentially 6 weeks, it looks like I think it's unlikely that everybody's going to follow everything. Nonetheless, we're going to carry on with translation today, which is the second half of protein synthesis. We're going to start off with a start on transcription. So there's an exam question on your screen now. It's 4 marks. It's a very standard story question. It comes up a reasonable amount in one form or another. So what I'd like you to do is I'd like you to, in a second, pause the video, get down just bullet points on this. You've already got some notes from your lesson yesterday, so get the bullet points down and make sure that you include every point you can think of. Just pause me now. Right, so as I said, this is a standard story question, so the mark scheme is predictable. If you're willing to learn that mark scheme, then you'll find that it will make your life a lot easier. So the mark scheme is as follows. There we are. Firstly, the hydrogen bonds between the bases are broken. Okay, the DNA bases. Then we have 3 RNA nucleotides binding to the exposed bases by the complementary base pairing rule. That complementary base pairing rule is important. You could also write A to C and A to U, G to C, and T to A. Remember that the RNA will have a uracil rather than a thymine. Only one strand is used as a template. That's actually a very important point. It's one a lot of people forget to write. If both strands are used as templates, that's DNA replication. It's not transcription. RNA polymerase binds together the RNA nucleotides or the new nucleotides. Do not accept bases for nucleotides is an important point as well. A lot of people write bases just by heart. They just don't even think about it. If you're writing bases, you're talking just about the A, the U, the G, and the C. And that's not what's joined together. The whole nucleotide is joined together. In fact, the bases don't get joined together at all. They just flip off by themselves. And then the pre-mRNA is spliced to form mature RNA. The average mark on this question is 3. Most people remember the hydrogen bond between bases being broken and the complementary base pairing rule and RNA polymerase. A lot of people will forget the template bit, number 3, and a lot of people will forget the splicing bit. So please make sure that you are getting on top of learning that. It does come up. But that's yesterday's lesson. So we're going to move on today with translation. Now, translation is probably the harder of the two, just because transcription is a lot like DNA replication, and so it's quite easy to remember, in my opinion. But translation is a bit more alien, so we're going to spend a lot of time on this. You're also going to see one of the finest examples of animation that any teacher has ever created, so you can look forward to that. That's coming up shortly. You need to make sure that you're keeping some detailed notes on this, but they're going to be from after the animation. I'm going to play the animation through twice, and we'll explain that in a second. So the aim today is basically to know what translation is and how different parts of translation are used. So the ribosomes, the ATP, and the tRNA. To describe the structure of tRNA is something we did yesterday, but on the syllabus it's put under translation, so you've already kind of done part 2. We will go back over that very briefly. And then the third one relates the base sequence of nucleic acids to the amino acid sequence of polypeptides. It sounds quite scary, but it's actually ridiculously easy. I'm going to show you how to do that in a second. So, usually we do this in a textbook in class. I don't think it makes much sense when we're sat there staring at each other for you to open your textbooks up. So, have a think about, what did we say the anticodon was yesterday? What did we say the anticodon was yesterday? And can you remember what I mean by small and large ribosomal subunits? So, just in your books, just write down the word anticodon. the title is translation if you haven't already. The word anticodon needs to be written down, and then can you remember what that is? And then the word small and large ribosomal subunits, can you write down what that is as well? You've done both of those in the past. We'll go over it after you play the video again. So, anticodon, first of all, this is something we talked about in last lesson, and it's related to tRNA, which is what you're looking at on the board. I hope you recognise that. So you've got your classic cloverleaf structure, and at the top, we're going to talk about later on, is where an amino acid will attach. We'll talk about that in a lot of detail in a second. And at the bottom is this anticodon. Now the anticodon is just 3 bases. They could be any base, they could be A, U, C or G, and they just stick out the bottom here. And so that's what an anticodon is. It's going to be very important, it's a key term. So you can say that an anticodon is a set of 3 bases sticking out of a tRNA molecule. And then we come to the small and large subunit. Well, this is relating to a ribosome, which is what you're looking at now. That's a ribosome. It's a drawing, obviously, not a microscope image. Now, as you should know from your cell topic, ribosomes are made of protein and RNA. They're made of a special kind of RNA called ribosomal RNA, or rRNA. You don't need to know that, you just need to know they're made of protein and RNA. There's a common misconception that the large bit is made of protein and the small bit's made of RNA. That's not a thing. Both of them are made of a mixture of protein and RNA, but we have to understand that it's made of 2 pieces, and it's made of protein and it's made of RNA. And that's as far as you need to know about ribosomes for the course. So, if we skip forward, we're going to start off, we're going to talk about the different stages of translation. I'm actually going to play you through this twice. The first time I just want you to watch and try and follow. The second time I'm going to slow down and we're going to take some notes and we're going to get it done properly. So, remember, this is at the end of transcription. We're kind of skipping out the splicing stage, which actually happens outside of the nucleus. We're imagining perhaps that we're in a prokaryote. So, splicing would actually happen just after the mRNA leaves the nucleus, but for now we're saying that we've finished transcription, we're on our way to translation. And the point of translation is to turn the RNA into a protein. So the RNA is going to go to a ribosome and it's going to be not turned into a protein, used to create a protein, used as a recipe for a protein. So, as I said, this is one of the finest examples of animation you'll ever see. I have to click it through manually. The mRNA leaves the nucleus through the nuclear pore and it goes into the cytoplasm. Once it's in the cytoplasm, it attaches to mRNA. Now I'll go over that in a second to be fair. So the ribosome attaches to the mRNA. And it attaches at a set point. A set point which is called the start codon. For what it's worth, if you go on to do any form of further biology, which you all will, we're talking about methionine is the particular amino acid. You may see that in your textbook sometime. But for now, we don't need to do anything about that. We just need to know it's going to attach at a start codon. Now, what do I mean by codon? I mean 3 bases on mRNA. So, there is 1 codon there, and there's a load of codons there. Every 3 bases is called a codon. Remember, I'm going to be going back over this in a second, so please don't scramble to write notes. Every 3 bases is a codon. Now, you might be able to start working out what an anticodon is then, but we'll go over that in a second. What's going to happen, this is a very simple mock-up of a tRNA molecule. Of course, in reality, it's cloverleaf shaped. The green bit is the tRNA, and the 3 bases at the top are an anticodon. So in this case, the anticodon on the tRNA says AAA. Now there will be tRNA molecules hanging around with all the different possible anticodons. AAA, UUU, CCC, GGG, GGC, and so on. I'm not going to go through all however many possibilities. But every single different anticodon type is hanging around. Now, each tRNA molecule is attached to a specific amino acid. So for instance, it might be that if you have an AAA anticodon, that will be a lysine amino acid set here, and that'll be a lysine amino acid attached to this tRNA. The blue blob here is the amino acid. The anticodon up here determines what amino acid will be attached. So if you have an AAA anticodon, you will have one amino acid. If you have a UUU anticodon, it will be a different amino acid. What happens is the correct anticodon with the correct bases which are complementary to the first codon will go and bind up by complementary base pairing, like so. It's attracted to it because it's going to bond with a hydrogen bond. So we've got a very weak hydrogen bond forming between the codon on the mRNA and the anticodon on the tRNA. And the amino acid is going to stick out the bottom. That blue blob is going to stick out the bottom. And all that's going to happen here is the same thing is going to happen again. So you get a second tRNA molecule, and I'm sure you could predict, looking at that second codon there, what the bases of that anticodon would be. So the bases of the codon are C, G, A. If the bases of the anticodon are C, G, A, then the bases of the anticodon would be G, C, U. So hopefully, yeah, there's my tRNA with GCU and its anticodon. Along it's going to come, it's going to do the same thing again. Now we've got two amino acids sat next to each other. That does not a protein make. We have to make a protein, and so we're going to have to attach those amino acids together. So we make a peptide bond between the adjacent amino acids. They're close to each other, some enzymes come along, which I'm not going to go into too much right now. Some enzymes come along, and they catalyze the creation of a peptide bond between the two amino acids. So now we've got two amino acids joined together, and this is the start of our protein. What happens now is the ribosome can only hold two tRNAs at a time. So the first one has to go away, the ribosome moves along, and the next anticodon attaches. And in this case, because we've got UGC on the codon, we have ACG on the anticodon. And so it will bring a different amino acid. And again, we form a peptide bond. And all that happens then is we get a repeated process of the tRNA breaking off, and a new tRNA coming along, and the ribosome moving along. So we're just going to do that over and over again. Now, eventually you will reach something called a stop codon. A stop codon is a codon which has a tRNA which doesn't have an amino acid attached to it. So in this case, the stop codon is CGC, excuse me. So there is a tRNA which should have the anticodon GCG, but this tRNA with the GCG anticodon doesn't have an amino acid attached to it. So we can't join anything on to this amino acid, and that is where the protein just stops. This polypeptide will then drop off and float away, because there's no more amino acids to attach. There's effectively a break in the chain. And then we have a polypeptide chain that's formed. What's going to then happen is it's going to float off into the cytoplasm, where it's going to be folded into the correct shape by various enzymes, which give it its tertiary structure. Now, a key point here is that several ribosomes can pass along the mRNA at the same time. A series of cars going along a road. So what you can create is multiple, hundreds if you want, of different of the same polypeptide, off of one mRNA chain. Now, that's one strength of this system. Because you can then make multiple mRNAs off of the same DNA gene, you can make many thousands of copies of a protein at the same time. So let's say I made 100 copies of the DNA into mRNA. I did transcription 100 times on the same gene, and each mRNA was translated. This is what this whole system is called, translation. Was translated hundreds of times. I can make tens of thousands of copies of this protein at the same time in literally seconds. So let's go over this again. I'm going to talk through it a little bit more slowly. I recommend you pause me at several points and take down these notes. Let's go right the way back to the beginning. This is quite complicated for some people, so don't stress if you're finding it difficult. Right. So first things first, the mRNA leaves the nucleus through the nuclear pore. Whether this is part of translation is questionable, but I wanted to link it back into transcription again. Then translation proper occurs. So you have loads of codons on here. Every 3 bases is a codon. The first thing that happens is the ribosome becomes attached to the mRNA. So that's your first note. A ribosome becomes attached to the mRNA. It's going to attach at the first codon, in this case UUU. And those codons are very specific. They are called start codons, though actually you don't need to know that for your A-level. Now, we then have a load of codons in here. And this is the second point, that the genetic code on the mRNA is read in groups of 3 letters called codons. So every 3 letters is a codon. Now that's a key term. It's not asked about as much as anticodon, which is asked about all the time, but it is a key term and it is important to understand. Then along comes a tRNA. Now tRNA molecules have specific anticodons for each of the 20 amino acids. So this tRNA molecule is going to come and attach by complementary base pairing to the first codon. And it's going to attach via hydrogen bonding. Right, so the complementary anticodon is attracted to the first codon on the mRNA and forms a weak hydrogen bond. Now, I'd like you to predict for me what would be the anticodon on the second tRNA. If you can get that, then you've kind of gone some way to understanding this whole point. So the second tRNA will have the anticodon GCU, and going backwards. There it is. This will then again bind via hydrogen bonding. The second codon on mRNA also attracts its complementary anticodon on the tRNA. Then we have a peptide bond is formed. This little blue line is what it represents there between the adjacent amino acids. And this is attaching the amino acids together because our aim is to create a polypeptide chain, eventually a protein, which we know is just a chain of amino acids. Then we have the same thing again. The first tRNA molecule has to become detached from its amino acid. That's the next point. And then along comes, next point, the ribosome moves along the mRNA molecule. And along comes the third tRNA. It moves in place the anticodon linking its complementary codon by complementary base pairing. And we have a peptide bond forming between the 2 amino acids. So we've got the third tRNA moving in, and then we've got a peptide bond forming. And from this point on, it's just a repeated cycle. This tRNA will now break off, the second tRNA will now break off, leaving behind the third tRNA. The ribosome will move along, another tRNA will come attached to this AUC codon, and you hopefully could predict that will be UAG. And on it comes, and there's no more notes from this point up until the very end, which is, I'll move my face up there, a stop codon is reached. the stop codon is reached. Now you don't actually need to know what a stop codon is or how it works, as long as you can say that it will stop when a stop codon is reached. Now, that's the end of translation. That's it. There is a final stage to protein synthesis. We wrote down our kind of set of 5 stages, including transcription and translation. The final stage is that it's folded into a protein, but we don't call that translation. That's a separate process. It's one we don't talk about at A level particularly much. Okay, that is ridiculously difficult. That's it. For a lot of people, that would be ridiculously difficult, particularly if you don't really get on with the chemistry side of things. Then this can be quite difficult. But the good thing is that there is a set answer for this. If you don't understand that, I do want to help you, but at the end of the day, if you don't understand it before your real A-level exam next year, then all you have to have done is memorized it. Because the question will be, describe the process of translation. Right, and then the final point here, which is important to get down in notes, is that several ribosomes may pass along the mRNA behind the first, each producing an identical polypeptide. So, assuming you've got down all those notes, and if you haven't, go back and pause me. We're going to move on to the next little bit on the translation topic, which is to talk about how ATP is used. Now, we've already completed the first learning objective, which is to explain the role of the process of translation. We've done the second one already last lesson. We need to be able to explain the specific roles of ribosomes. I feel we can probably have a go at. tRNA, yes, but ATP. Now, ATP, as I'm hoping you remember, is a chemical which is used to release energy, and it's made during respiration. And what it's going to be used for here is to what we call activate this tRNA. And what we mean by that is simply attach an amino acid onto the tRNA to create something that looks like that. If you think about it, at the end of the last bit of translation, you are left with a polypeptide chain, which is fantastic. You have the ribosome, which will float off and do its own thing, and you're left with one of these for every single codon in the mRNA. It used to have an amino acid attached to it. That amino acid has been used to form a polypeptide, and now it's left with just an empty tRNA. And so we need to add an amino acid onto that. And the way we do that is by using ATP. We convert it to something called AMP, but you actually don't need to know that. That's just adenosine monophosphate, so it loses 2 of its phosphates. But all you need to know is we're going to be using ATP to attach this amino acid onto this tRNA molecule. That's what ATP is used for. So what I'd like you to do now, I'm going to stick, and perhaps you could take down those notes, that might be useful for you. I'm going to stick a table on your screen, which is basically going to ask you for each of the 3 important molecules we need to know about, what they're used for in translation. So here's your table. ATP, tRNA, and ribosomes. I'd like you to, in a second, pause me, draw this table, and fill in for each molecule type what its role in translation is. Hopefully you've done that. Now, what you should find here, ATP we've just talked about, is going to be used to attach the tRNA to the amino acid. tRNA is used to bring the amino acids to the mRNA in the ribosome. And ribosomes, perhaps the hardest one to define, they're simply binding sites for the tRNA and the mRNA. You can think of them almost like giant enzymes. They're not in any way enzymes, let's be clear about that, but you can think of them a bit like that, because what they're doing is they are providing a site where the tRNA and the mRNA can both bind on and effectively be held in place until this chemical reaction, the formation of the polypeptide, of the peptide bond, has occurred. So they are providing a binding site for the tRNA and the mRNA in order to facilitate translation. Right, this last bit is not too difficult, I wouldn't say. We're looking at how we're going to know which anticodons are going to hold on to which amino acids. So there are, as you can see, a number of different possible combinations. Now, there are 64 possible combinations for only 20 amino acids. And what that means is there is multiple possibilities for each amino acid. For instance, phenylalanine at the top is coded by both UUU and UUC. So if on your mRNA it says UUU, on your codon, you will get phenylalanine. If it says UUC, you will also get phenylalanine. If it says UUA, you will get leucine. We'll talk more about that in a second. What I would like you to do, just have a glance at this, make sure you can follow this table. What amino acid would you get if your codon on your mRNA was UGU? So hopefully we're all looking at, there's the first position U, there's the second position G, third position U, UGU is cis or cysteine. You can also see that there is a set possible combination which will make up a stop codon. So if at any point you have a UAA hanging around, that will end the protein right there. We're going to make sure you can use this. What I'd like you to do is get your table, which I've attached one of these into the assignment, and I'd like you to use it to do this question. Now, I've kind of picked this out of a different PowerPoint. I just want you to do the first one. So you've got a set of bases here, which are DNA bases. And I'd like you to tell me what the mRNA strand will say, what the polypeptide chain will be, and how the mutation, no, forget number 3, I apologize. So just for the first strand here, just do the first 2 questions. So by the end, you should have an mRNA list of bases, and you should have a list of different amino acids. So pause me there. So that's a template strand, which means the mRNA will be complementary to that strand, because the bases will attach to that strand. So the first 3 bases on your mRNA will be AUG. And AUG, if you flick back here, will be methionine, and it is down here. The next 3 are AAG, and again, AAG would stand for lysine, LYS. Then we have CUA. CUA is leucine. AAC, which would be ASN or aspartame, and then CAC, or in this case we would go for GUG, because of course it has to be complementary, and GUG is valine, VAL. And then the last 2 will be almost leftovers. There's not a full code on here, so we can ignore this CT on the end. So if you got AUG, AAG, CUA, AAC, GUG, GA, and the correct amino acids as I just described them, then you've understood this little bit. It's important to note, firstly, that you will never be asked to memorize this table. That's very important. You will be given this table if you need it, or more usually, a very small part of this table. So don't worry about that. Right, there is a sheet in the assignment which is about protein coding. If you feel like you're struggling with that a little bit, or you fell behind a little bit, open that sheet up and have a go at it. And it's basically a load of these, and I want you to have a go at them and see if you can do it. If you can, then you will have no issues. I don't have any problems with people just doing 1 or 2 of them. I'm not going to take this sheet in. It's there to make you confident that you can do this. Now, the next bit we're going to talk about is a couple of terms which we need to know for the genetic code. So we're going to have a look at this slide. Now, this slide tells you some features of this code. Some of them are very obvious, but there's some terms in here which you get asked about a lot, which you do need to know. The first thing which we can say about the triplet code is it is universal. What I mean is that every living thing that we are aware of, certainly every living thing we've ever come across, uses exactly the same code. Now that is incredibly important. Firstly, it gives us a hint as to the idea that we are all related to each other. But most importantly for modern medicine is it means that if I put a human gene into a bacterium, it will make the correct protein because it uses the same code. This is used for things like insulin. Insulin is made by bacteria rather than pigs as it used to be, because if you put an insulin gene into a bacterium, it will make correct insulin. If we had a different code, if they said that instead of TTT up here, which is the DNA code making phenylalanine, it made leucine, then obviously it wouldn't work. Non-overlapping. What that means is that there is the TGT, top right, always codes for cysteine and never for tryptophan. There is only one code, there's only one amino acid for each code. Degenerate means that there can be more than one code for each amino acid. So, for instance, leucine up here has 6 different codes, near the top left, has 6 different codes, all of which will make leucine. They are degenerate. Always read in the same direction, which means that you always read from 5' to 3'. You can't read backwards. The start code always being ATG. Actually, I wouldn't worry too much about that. It doesn't come up and I don't really need you to know about it. And the fact that the stop codes exist, that they are a thing. Now the ones you absolutely need to know are degenerate and universal. They come up a lot because people forget what they mean. Universal means everything has the same code, all organisms. Degenerate means there's more than one code for each amino acid. So, in your books, can you get definitions down for these 3, please, if you haven't from what I've been saying? What do those 3 mean? Degenerate, non-overlapping, universal. So hopefully you paused me. We said degenerate means there's more than one code for each amino acid. Non-overlapping means there's only one amino acid for each code. Each code only codes for one amino acid. Universal means all organisms have the same coding system. Fine. Now, what we're going to talk about now for the last bit, actually we're going to move on just to an exam question to finish. So here's the exam question we're going to finish off with. It's 6 marks and it's about translation. I'd like you to read through it and complete the question. You have 6 minutes, just pause me and you can get on with this. So hopefully you now have an answer in your book of what the process of translation is, and I want you to have a think about this tomato plant. It's a very interesting question. The first one is quite a boring standard story question. It's worth noting this comes up quite a lot. Describe the process of translation. And there's 4 marks here. mRNA attaches to ribosome. You get a mark for that. Codon on mRNA. You get a mark pretty much for writing the word codon. Binds to anticodon on tRNA. Each tRNA brings a specific amino acid. I wouldn't necessarily expect everyone to write that the sequence of codons determines the order of the amino acids, but that is true. And then the formation of peptide bonds by condensation reactions. So there are, I believe, 5 possible marks. I know 6. So hopefully everybody got their 4. Now this next one is very interesting. It's about this softening gene. And first of all, you have to notice something very important about the softening gene and the inserted gene, and that is that they are complementary. Every A goes with a U, every U with an A, C with a G, G's with a C. Now you have to think about why that might be important. Why do I care that they are complementary? And the answer to that question is not that it will create some kind of backwards anti-softening enzyme. That's not a thing, unfortunately. Reversing the order of some bases does not create the opposite protein. It creates no protein. So if we tried to translate that inserted gene mRNA, we probably wouldn't create anything of any use at all. But what would happen if I put that mRNA inside of a cell with a load of softening mRNA in it? Hopefully, what we would see is that it would automatically form hydrogen bonds between the U's on the inserted gene and the A's on the softening gene. And so the marks are, inserted gene is complementary to normal gene. And you get a mark for that. That means even if you can't figure out why that matters, having noticed that gets you a mark. And with difficult questions like this, it's worth writing down just anything you notice, because it's often worth a mark. That it will bind to it to prevent protein synthesis. That the mRNA will not be able to bind to the ribosome very broadly. And in this third part, I don't think anybody would ever write. I know I wouldn't have. It's not necessary because you can get 2 marks without it. But the fact that it won't stop all translation as some mRNA will reach the ribosomes, that is true. That is the case, but I can't see how it's relevant for the question, to be honest. Now we're going to cover something a bit more like this in year 13 when we do some gene technology work, but hopefully everyone at least noticed that it was complementary. Right, if I'm not wrong, then that will be the end of the video. Now, hopefully that everyone understood everything there is to understand there, but what we need to know by the end is this. You need to explain the process of translation, explain how ribosomes, ATP and tRNA work, describe the structure of tRNA, you should already be able to tell me, and relate the base sequence of nucleic acids to the amino acid sequence of polypeptides. Basically, if you're given that table, can you use it to tell me what amino acids there will be there? And I want to add on to there some looking at the words degenerate, universal, non-overlapping. If you can do all of those things, then you've got this topic sorted out. Well done, ladies and gents. I know it's been a long one, this one. Hopefully you've been able to get it done in the time, but if you have not, I will try and make the next one a short one so you have time to fill it in.